So this is the last video and last part of the lecture on cellular respiration here. And like I told you, we were going to talk a little bit about what happens with some of the other foods that we eat other than glucose, and also a little bit about how cell respiration is controlled here and regulated, right? So this is cell respiration that we have learned about already, so I don't need to go back through all the stages here. But we've focused solely on glucose before. But we eat other things. Man, would it be boring if all we did was eat glucose all the time? I mean, ugh, right? I mean, I love eating all kinds of different foods, and I would hate to just eat glucose, but then my body has to be able to handle all the different kinds of foods that we eat. So to start with, glycolysis accepts a wide range of other carbohydrates as fuel, not just glucose, right? We eat polysaccharides, pasta, bread, and all of those other starches. And our body then breaks all those polysaccharides down through hydrolysis to turn them into glucose, right? And then other 6-carbon sugars that we eat, if we eat fructose, if we eat galactose, any other 6-carbon sugars that we eat are modified with different enzymes and converted into glucose, right? So like I said, galactose and fructose. But what about proteins, right? We know we're supposed to eat proteins daily. And what happens with proteins when we eat them is enzymes break them down into their individual amino acids, right? And all amino acids, we remember, have this basic formula with an amino group, a carboxyl group, and then the central carbon that has a hydrogen and some kind of R group, one of those 20 different R groups attached to it. So this 2-carbon fragment here is going to be removed and either enter glycolysis or the Krebs cycle, depending on what this R group is, right? The amino group is removed and becomes part of our urine, right? It gets converted into urea in our bodies. Other animals convert it into different nitrogenous waste products. That's why it's yellow, highlighted in yellow here, right? The amino group becomes waste, while the rest of the molecule will either go to glycolysis or the Krebs cycle, depending on what this R group is here. Fats, we eat plenty of fats. I'm sure over Thanksgiving weekend, everybody had their fair share of fats. So what about fats? How do those get handled, right? Well, first of all, fats are going to undergo hydrolysis into glycerol and fatty acids, right? That glycerol is a 3-carbon compound that's converted to G3P, one of the intermediate molecules of glycolysis, if you remember. And that G3P is shuttled to glycolysis and enters glycolysis at that 5th or 6th stage there. Okay. The fatty acids go through a process called beta oxidation and are broken up into individual 2-carbon fragments, okay? And those 2-carbon fragments then are going to be shuttled off to the Krebs cycle and attached to coenzyme A and then enter as acetyl-CoA, right? Interesting to note that fat generates about twice as much ATP as carbohydrates because we have so many more carbons in a gram of fat and more energy releasing bonds. We also have a lot more of those carbon-hydrogen bonds to be oxidized. There's also more oxygen in a gram of carbohydrates. So carbohydrates are already partly oxidized. We've got a lot of not even partly oxidized bonds right here. Okay, so there's more energy to release in fat. So our metabolism is the sum total of all of our digestion reactions, the catabolism that takes place in our bodies to provide us with energy and the raw materials for synthesis. And so metabolism is the sum total of all the digestion and synthesis reactions that occur within our bodies, okay? And it's controlled by regulating enzymes through feedback mechanisms where when we've got a lot of the raw material, that stimulates synthesis, and when we're running out of energy, that stimulates digestion, right? And then once we have enough of our synthesis products, those products inhibit further production of themselves, okay? So when we talk about digestion and we're talking about the digestion of carbs, fats, and proteins, they all get catabolized through the same pathways, but enter at different points, right? So carbohydrates are going to enter here in glycolysis at one of our stages of glycolysis. Um, So the cell is going to get energy from every possible source here. We talked about the amino acids having the amino groups removed, and then depending on what's left, they can enter into glycolysis. They can enter as acetyl-CoA, or they can be converted into one of the intermediates of the Krebs cycle. And with fats, we talked about glycerol being converted into G3P, and the fatty acids broken into 2 carbon fragments and entering as acetyl-CoA. So all these different molecules are catabolized and then enter our cell respiration pathway at different points. In terms of synthesis, when we have enough ATP around, then we can use points in glycolysis in the Krebs cycle as links to run these pathways backwards. So we can say take that G3, if we got plenty of energy, so the cell can siphon off some of the G3P and convert it back into glycerol, and some of the acetyl-CoA, convert those back into fatty acids and build fats, right? The cell can take some of that pyruvate and convert it back into amino acids, right? So we can run these pathways backwards, okay? Once we have enough fuel, then our body can convert some of what we've eaten. If we don't need any more fuel and we have plenty of ATP, a lot of what we eat is going to get converted into fat then. Okay, so the real key point for this particular lecture, though, is how the cellular respiration is regulated here. And it's regulated through feedback mechanisms, right? And the way that this is done is that we have this multi-step pathway. This one happens to be require 6 enzymes, this particular pathway. Okay, and when the final product accumulates in large concentrations, it feeds back and serves as an allosteric inhibitor of an enzyme earlier in the pathway. Okay, that way, once the cell has enough of this final product, it doesn't unnecessarily accumulate it. It doesn't waste its resources in producing more of it. Okay, so the product is self-limiting. So the key point of control in our cells, okay, I'm going to put this over here, put my video over there. The key point of control for cell respiration occurs at that third step with the enzyme, my favorite enzyme, phosphofructokinase, right? And this enzyme, phosphofructokinase, is allosterically regulated. The reason that the regulation occurs here is the step that it catalyzes is the can't turn back step before splitting glucose. Once phosphofructokinase adds that second phosphate group onto our glucose molecule, it is going to split and the rest of respiration is going to take place. So this is a good place to stop things. It's really the last place you can stop things in glycolysis. So when AMP levels and ADP levels are low, when AMP levels and ADP levels are high, that means ATP levels are low and the cell needs to make more ATP. So AMP and ADP bind to phosphofructokinase and activate it. They convert it into its active conformation so that it will continue to function in phosphorylating, adding that second phosphate group onto glucose so that cell respiration continues. Okay, ATP, when it's in high concentration, then that means the cell has enough ATP and is not using it as fast as it's being produced. ATP actually binds to phosphofructokinase and it allosterically inhibits phosphofructokinase, which means that none of the rest of the steps of glycolysis take place. We don't even generate the ATP via substrate level phosphorylation from glycolysis. It also means that the Krebs cycle and the electron transport chain don't take place. Citrate also feeds back and inhibits phosphofructokinase. Because if citrate levels are really high, that means that the Krebs cycle is producing lots of citrate that is not being used. And so that's kind of a way to coordinate the Krebs cycle with the electron transport chain. Okay, so that is where I'm going to stop here for today. If you have any questions about anything related to the regulation of cell respiration via the allosteric enzyme phosphofructokinase, please ask during our review session or sign up for an academic support time. So that's all I have for this lecture. Have a great day, and I will see you next class.